Good afternoon. My name is Catherine Cunningham, and I'm the chair ad interim of the Department of Pharmacology and Toxicology, and also the director of the UTMB Center for Addiction Research. I'd like to welcome you to our annual Chauncey Leek Memorial Lectureship. This year we're coupling that with a poster session and lunch after the seminar that you're also invited to. I'd really like to welcome the members of our Galveston University community, the local community of individuals who are very interested in this topic, and our colleagues and friends from universities in Houston, San Antonio, and as far away as Lubbock. So we're glad to have you here today. We're also very lucky in that we have a slate of individuals we'd like to warmly welcome with a special hello. And these are local and state representatives that have joined us to see Dr. Volkow. We have esteemed U.S. Representative Craig Island, I believe, here. I don't know. I can't see very well. We have Honorable County Court Judge Mary Nell Crepito. Thank you. Honorable County Court Judge Roy Quintanilla. Honorable Judicial District Court Judge John Ellisor. Honorable Justice of the Peace Jim Schweitzer. And we have Honorable CPS Judge Suzanne Schwab-Radcliffe, who I didn't see come in. There she is. And Galveston County District Attorney Kurt Sistrunk had intended to be here, and I believe Donna. Is Donna here? There she is. As Assistant DA, if I'm correct. Thank you for joining us. We're really appreciative that you've come today. And I know everybody in this room is interested in solutions to drug abuse and addiction. And that's what we share. It's a major societal problem that needs to be fully tackled. And Dr. Nora Volkow has devoted her life to finding solutions through science. Today we honor Dr. Volkow as our Chauncey Leek lecturer, and just a minute to tell you that Dr. Chauncey Leek holds a special place in the hearts of pharmacologists and also UTMB pharmacologists and scientists. He was an internationally known pharmacologist, philosopher, author, and linguist. And he served as the executive VP and the Dean of the School of Medicine from 1942 to 1955. So he's a big part of our past. This lectureship is actually funded by his sons and their families, and we're very pleased to have the opportunity to invite Dr. Volkow. Dr. Volkow has a very interesting history. She grew up in Mexico City as the great-granddaughter of Leon Trotsky, the revolutionary, and spent received her MD from the National University of Mexico in Mexico City, moved to the U.S. to complete her residency in psychiatry at New York University, and in fact moved back this way for her first faculty position as assistant professor of psychiatry at the University of Texas Health Science Center in Houston. So Dr. Volkow spent 3 years as a naturalized Texan, and she was then recruited away from Texas to Brookhaven National Laboratory in SUNY Stony Brook, where she marched rapidly through the ranks to become chair of the Stony Brook Medical Department and associate dean of medicine at SUNY. In 1993, Dr. Volkow was appointed director of the National Institute on Drug Abuse, which is an NIH institute that funds over 90% of the world's research on abused drugs, and that's pretty significant. Dr. Volkow is passionate and committed to the mission to cure addiction. Her 25-year career is marked by a multitude of groundbreaking studies which have greatly advanced our understanding of human behavior, motivation, and brain chemistry. Amazingly, she has published over 440 peer-reviewed publications. She's an editor on 3 books and more than 80 reviews and book chapters. And I think what's really, truly amazing is that from 1993, when she took over this really sizable new leadership position. Over 150 of those peer-reviewed publications have occurred since she took that role. So she's sustained a very active scientific career. A member of the Institute for Medicine and a nominee for National Academy membership, Dr. Volkow has won many, many awards, including she was named Innovator of the Year for 2000 by U.S. News and World Report. In essence, Dr. Volkow is changing the way we think about drug abuse and addiction and is at the forefront of developing new science-based prevention and therapy approaches to substance abuse. So, on behalf of the UTMB Department of Pharmacology and the Center for Addiction Research, I'd like to present Dr. Volkow with the Pharmacology Department's highest honor, the Chauncey Leake the Chauncey Leake Memorial Lectureship Award. Nora? And now I'd like to welcome Dr. Volkow to talk about her research, addiction, conflict between brain circuits.
First of all, good afternoon, everybody. It's a pleasure to be here. I have some viruses circulating to my body, but I'm actually in conflict with them. But it's a pleasure to be here in Texas, to be here in Galveston, and I want to thank the school for having given me this opportunity. But I specifically do want to thank Kathy Cunningham for her perseverance, because my trip got diverted by the hurricane. I was coming and says, no, Nora, we have the hurricane, you cannot come. So the hurricane did not allow me to come, and then I was coming, and then I had to attend a service with Obama, and I was told, I think like 1 week before, so I called Kathy and says, Kathy, I really feel embarrassed, but I have to do this thing with Obama, for which, by the way, I was very excited about. So she said, okay, Nora, we'll plan January. So I was feeling sick. I said, no way, I'm coming and I'm here. But I want to thank Kathy, not just for her perseverance on bringing me here. By the way, yes, I was 3 years for Texas, but I got so attached to Texas that this is not something I've told Kathy, actually. I've not told many people that I've actually kept my apartment in Houston because I just couldn't let go. So that just get you a sense about how much I like Texas. Now, but I want to thank Kathy because one of the things from the inception of her career, I've been attracted by her work because she does not follow the path followed by everybody. And her work attracted me immensely because she was looking into the serotonergic system vis-a-vis its involvement with the process of drug abuse and addiction. And very importantly, how can we use that system to try to develop new ways of treating it? And she has been unique in doing that and persevering against the whole current of investigators that have been focused, like me, on the dopaminergic system. Now, it's not that I have not been interested on the serotonergic system. I've been very interested on the serotonergic system. I think it's extraordinarily relevant. I have not been able to tackle it because I do not have had the ligands that would have enabled me to look at it in the human brain. Had I had them, it would be from my inception into the whole field of brain imaging to understand what is the neurobiology underlying the disrupted brain of a person that's addicted. It's been very clear in my brain that the serotonergic system is involved, and Kathy has paved that way with her elegant preclinical studies. So I want to thank her for inviting me here, but also for her vision in terms of research into the whole field of substance abuse. It's also been a pleasure to actually get to interact with you. And I was in Houston, so I really didn't know much about the medical school in Galveston. And even though my trip has been very short, I've been actually very impressed with what I've seen. And I look forward to expand the collaborations. I always say my job at NIDA, which actually, no, I didn't start 1993. I started 2003. I've been in this job for 6 years. It's the only factual error, is that my job is easy, theoretically, is if I get the best brains to actually think and invest their energies and creativity into fighting the problem of substance abuse, I've done my job as a director. So the opportunity to try to actually participate in coming here and identify ways in which your research is very, very relevant for the substance abuse field is important as one of the tasks that I do as director. So, and I hope I hope that there will be an expansion of these collaborations. Now, let me get to the essence of the talk, addiction conflict between brain circuits. I've been very intrigued since I was a medical student about why is it that people that become addicted to drugs lose basically almost complete control of their actions, even though they cognitively no longer want to take the drug, and even though the consequences are actually quite catastrophic. So this dissonance in terms of the inability of a person to say, I do not want to take that drug anymore, I don't want to end in jail if I continue taking, or lose the custody of my children, those very powerful negative reinforcers are insufficient to control the behavior of a person that's addicted. Now, to me, that was an opportunity to go inside the human brain and try to understand what are the brain circuits or systems that enable us to exert free will. Because in drug addiction, it has been dismissed for many, many years as, oh, it's just a problem of free will. Dismissed as it's a problem of free will. But free will is the product of our brain. And by focusing on the function of the addicted brain, one can, of course, gain knowledge about what are the systems that enable us to exert free will. And when we say, no, I don't want to do this, we're able to inhibit those actions. Now, where do we start? I said our work focuses on the dopamine system, not because it's the only system involved, but because it's a very important one, but also we had the tools to directly investigate in the human brain. Now, why the interest on the dopamine system? The interest came around because all of the drugs of abuse, whether it's licit or illicit, all of them increase dopamine in the nucleus accumbens, this area here, which is considered to be one of the main pleasure or reward centers of the brain. The ability of drugs to increase dopamine is brought about by different molecular mechanisms, some direct, like the case of cocaine or amphetamine, and some indirect, like the case of morphine and nicotine. But all of them increase dopamine in the nucleus accumbens. And you see this in this study in which you actually, in a rat, you can put a little probe inside and you measure changes in dopamine as a function of the administration of amphetamine, and you see the very large increase. Now, this circuit was not in our brain or in the brain of rats or many other species. Basically, all mammals have this system for us to take drugs. This system of reinforcement or reward is hardwired in our brain. So it is biologically normal and expected that behaviors that are pleasurable are going to increase our motivation to repeat them. And this is the mechanism, for example, by which nature ensures that we will do behaviors that are crucial for survival. What are those crucial? On the top of the list, I would sort of say food. I would sort of say sex for procreation. And of course, the concept of the nurturing of the mother into the infant. Crucial behaviors. So in order to motivate and drive the behavior, you have to have a reinforcement. So these circuits were generated by nature to ensure that we will be doing the behavior appropriately. And indeed, when you are food deprived, for example, and you put food in front of an animal, you see the increase of dopamine in this brain area. And this increase in dopamine, the magnitude of the increase, will actually predict how motivated the animal is to work in order to achieve that food. What is interesting and what drugs have done is basically in randomness of nature that there are these compounds that can directly go into the system and literally hijack it, go directly there and disrupt it. And so as a result of that, you have some consequences that are very adverse. Let's look at the system with food. Nature is incredibly clever and has an enormous amount of wisdom. So you need food in order to survive, and dopamine goes up if you're food deprived because you need it. But once you've satiated yourself and that food is no longer necessary, unless it's certain types of food like chocolate, which don't behave properly, then the brain, the nucleus accumbens, no longer increases dopamine with the food. And as a result of that, you no longer have the motivation and we stop eating. As I say, this is different for chocolates. But what happens with drugs is because the drug directly, by its pharmacological effects, is increasing dopamine, you do not get this tolerance effect. And so you can take the drug, and dopamine goes up, and you take it again, and up and up. So there's no satiety. And that drives the compulsive behavior that is so characteristic of drug taking. Now, the other difference with respect to the natural mechanism versus the unnatural chemical one is look at the magnitude of the effects. The increases in dopamine when someone takes a drug like amphetamine are much greater in magnitude and in duration than those of food. So it's what we call a supraphysiological stimuli on a biological system. And when you do that, what you generate is adaptation processes that try to compensate for these supraphysiological responses. And the consequence is that when you are no longer on the effect of these drugs, and this has been shown in animals, these animals, when they don't longer have these maximal increases in dopamine, because they've adapted to this, they are no longer responsive to natural reinforcers. And you need much greater stimulation to activate the same system. So in a way, they are paying long-term consequences for the immediate reinforcement. Now, this is in animal studies, and the question is, do similar things happen in humans? That is to say, that drugs increase dopamine in this area of the brain that is so important for reinforcement and reward. Well, we are able to now do these studies in humans. We are not putting a cannula, of course not, but we're using imaging technologies, and we measure it indirectly. And it's a very simple method, the way that we do it. We take a radioactive substance that has a very low concentration of radioactivity, very short half-life, 20 minutes, that specifically is attached to a compound that will bind to dopamine D2 receptors. But it can only bind to these receptors when the dopamine, which is the signaling molecule, is not occupying them. Normally, when we are currently like now with no drug effects or no novel stimuli, 85% of our receptors, or 90%, are free. So you can inject your C11 raclopride. C11 is the isotope. And you can see and then take an image, and you see the accumulation in these areas of the brain here, which is where you have the nucleus accumbens located. And very much that radioactivity represents the raclopride binding to the dopamine D2 receptors. And the method that follows is you can give the subjects, for example, a drug, intravenous cocaine or methylphenidate or nicotine, whichever drug you want to do. We use intravenous methylphenidate in our studies because it is a drug that is used therapeutically, but curiously, as a pharmacological agent, it is very similar to cocaine. So just like cocaine and a similar affinity for its target, methylphenidate blocks the transporter, and the dopamine transporter's functions in the dopamine cell is to basically, when dopamine is liberated, it basically stays in the synapse, in this space, for a very short period of time. So it has a very finite amount of time to interact with the receptors and send a signal, which is the one that is reinforcing. When you block, because it's immediately picked up and recycled into the cell through this transporter here in red. Cocaine blocks this transporter, and by so doing, dopamine can no longer be recycled, and it accumulates, and that amplifies the signal. And it is now understood, well understood, that the ability of cocaine to specifically block the transporter is crucial for its reinforcing effects and accounts for its high addictive potential. Interestingly, methylphenidate is also blocking the dopamine transporter for a similar affinity, actually slightly higher. And when you do that, then, of course, dopamine can no longer be recycled, and it accumulates, and it occupies these receptors. So then, using the raclopride molecule labeled with carbon 11, you inject it, and you get an image of the brain. And then you can compare the images when the subject had a placebo, no drug, versus when they have the drug, in this case, methylphenidate. And with appropriate mathematical modeling, by comparing the binding under this condition versus this one, you get an idea of the relative changes in dopamine produced by that particular drug. And when you do that, the subjects are awake. You can actually see that this is the % change in dopamine using this method versus the subjective reinforcing responses to the drug, since you ask the subjects how high or how euphoric you feel. And we and others have reported this particular finding that, as would have predicted from the animal literature, the higher the changes in dopamine, the more intense the reinforcing effects. Now, we're indicating, indeed, that the ability of drugs to increase dopamine in humans is crucial for their rewarding effects. However, one of the things that was very intriguing in all of these studies was in this particular investigation, in order to mimic the effects of intravenous cocaine, we had to give methylphenidate intravenously, and cocaine abusers cannot differentiate intravenous cocaine from intravenous methylphenidate. But at the same time, in my brain, I was questioning, well, you know, we give methylphenidate to little kids. It's the #1 most prescribed drug in children in the United States. And I said, well, how can it be? And of course, we don't give it injected, we give it orally. So I was curious to know then, well, what happens when we give it orally? Do you get a high, and do you change dopamine? Well, what we found is here, this is oral methylphenidate. In this case, the measurement was done, the dopamine changes within 1 minute, and in this one, we give oral methylphenidate and we wait 1 hour. And that's because when you take a pill, it takes a long time for the pill to get into your brain. And whereas when you inject it, it goes immediately. So that when we do that, we first saw the question, does it change dopamine? And indeed, it changed dopamine basically in comparable amount to this dose of methylphenidate, which was 0.5 milligrams per kilogram. But what was interesting was that even though we were changing dopamine, we were not producing a high. And so here you have these apparent discrepant results that where you see an association between the ability of the drug to increase dopamine and the high, and in this one where you're increasing dopamine but you're not seeing a high. So why is that so? Where is that discrepancy coming along? And what it's telling immediately in my brain, of course, I have the story of doing the experiment, so I've thought about it a long time, so it's not automatic. A main difference is the dynamic nature of the changes. In this one, the changes in dopamine are occurring within 1 minute, and in this one, the changes in dopamine are occurring with 60 minutes. And it does become clear in biology, though we don't pay so much attention to it, that the signaling, the way that neurotransmitters signal, is not just dependent on the magnitude, how big the changes in dopamine are, but importantly, how fast they are occurring. And this starts to make sense in a way, in an interesting way, because in animal literature, it was understood that the rapidity at which a drug gets into your brain is a crucial variable that determines its reinforcing effects. So the faster it gets there, the more intense the euphoria. And this explains why the route of administration by which you get a drug has such a big impact on its reinforcing effects and its addictiveness. So say, for example, if you snort cocaine, the likelihood and probability of becoming addicted is much lower than if you smoke or you inject it, because the drug is a much more potent reinforcer. So this could explain, indeed, why when you are giving oral methylphenidate, you are not getting a high, because the drug is taking very, very long. So we were intrigued by that possibility. So we went ahead and actually, to try to look further into it, we did a study where we, these were, of course, crucially involved with a chemist, where we label cocaine with a carbon-11. This is the radioisotope with 20 minutes that you measure with PET. And we labeled methylphenidate with a carbon-11 molecule. And because you are substituting what carbon with a carbon-11, you actually literally have the drugs. And you can use PET to monitor not just where they go in the brain, in the human brain, but actually the duration of those effects, the temporal sequence of those effects, which in pharmacology we call it pharmacokinetics, which is actually basically being able to do with this technology what sounds like science fiction, which is pharmacokinetics directly in the human brain. And this is what you see here. So you see C11 cocaine, C11 methylphenidate. Fair surprise to us, the distribution was almost identical. Look at this distribution here at 8 minutes versus 10 minutes. Very, very similar distribution. They are binding actually to the same sites. How do I know? We pre-treated subjects who were given the radioactive cocaine with pharmacological doses of methylphenidate, and we blocked the binding of cocaine. And we also did the opposite experiment. I keep on touching this thing. The opposite experiment where we, this was done in cocaine abusers, where we gave them cocaine, pharmacological doses of cocaine, before giving them the radioactive methylphenidate, and then methylphenidate could not bind in the brain. So we have two drugs that have similar distribution, similar binding. But we use one, this is highly addictive, and this one, though it can produce addiction, it rarely does. So what was driving that? So here we have the distribution very similar, but the pharmacokinetics very different. First question, which was not evident even then. The cocaine gets into the brain very rapidly when you inject it intravenously. You are peaking at around 6 minutes. Intravenous methylphenidate peaks very rapidly. You are peaking a little bit later, 8 minutes, 10 minutes. But for practical purposes, you get a huge amount of methylphenidate very rapidly, which is the relevant factor for reinforcement. So you see at 6 minutes, you get, or you can already predict there's a lot of activity there. But there's a very striking difference between these two drugs, and that's the rate of clearance. Cocaine gets out of the brain very rapidly. By 40 minutes, there's almost no cocaine in your brain, and there's almost nothing binding to the transporters. In contrast, at 40 minutes, approximately 60 to 80% of the methylphenidate is still bound to the dopamine transporters. So the clearance rate is much slower. Now, we were a little bit confused by this because theoretically, if we had just assumed the ability of drugs to increase dopamine is what leads to the reinforcing effects, then it would have followed that these drugs should have been more addictive than this one, because this one has such a short effect, whereas this one will be increasing dopamine for a long period of time. But when we query the system to try to understand these discrepancies that were not making logical sense in the brain, you go into the data, and that's why you actually do the experiment. And what you're going to see here is the diagrams that transform these data from an image into the activity in the part of the brain that contains the nucleus accumbens. And in these same subjects, you are actually monitoring also the temporal course of the high. So we're looking at the concentration as a function of time of cocaine in the reward centers of the brain versus the subjective perception of the subjects for the high. The drug in this plot is in dark blue, and you see the drug gets into the brain, cocaine very rapidly, peaking 6 minutes, and then it goes out of the brain very rapidly. The temporal course of the high follows the very fast uptake of the drug in brain, peaking at 6 or 8 minutes, and the high returns to baseline very rapidly, and by 20 minutes you basically have no effect of drug. And these two go very much in parallel, the fast uptake with the intense fast high, and then this one goes back to baseline, perhaps a little bit faster, but for all practical purposes, very similar. When you do that same analysis with intravenous methylphenidate, you get a completely different picture. While the initial very fast high is similar to that of cocaine, as the drug gets into the brain very rapidly, you get the intense high. You see that the high from methylphenidate returns to baseline very, very rapidly, even though the drug is still bound on brain. So you see that dissociation. And what in my brain is telling you is that if you look at this data and you try to extract what could it mean, in my brain, it basically means that it is the fast change in dopamine that is associated with a high, but not necessarily the steady state increases in dopamine. And that, of course, could exactly explain why, in this case, you get a high 1 minute after administration, but not in this case, where 60 minutes, the dopamine has maintained itself steadily going up. So you need the disruption of the dopaminergic system in order to be able to generate a high. Now, of course, this makes a lot of sense if you know what the neurobiology of the dopaminergic pathways are, because the dopamine cells, which are the ones that are liberating dopamine, function in two types of firing. One of them is the phasic dopamine cell firing, which leads to very fast changes in dopamine. And it is this fast dopamine cell signaling which is associated with reinforcement and saliency. So the natural response of the dopamine cells when something is salient or reinforcing occurs with this type of very fast dopamine, very short-lasting but very fast, that increases dopamine very abruptly. In contrast to another mode of this dopamine cell firing, which we call tonic dopamine cell signaling, that maintains much lower levels but steady state levels of dopamine that, on the other hand, are going to determine the preparedness, the readiness of the system to respond to external stimuli. So these two modes of operations, even though they are using the same chemical, have very different functions. basic dopamine signal in order to be able to respond immediately and gather the attention and the drive and motivation of the organism, whereas the tonic dopamine cell signaling to maintain that behavior and that receptivity. So then it then doesn't surprise what drugs are doing. It's not just basically increasing dopamine, but their ability to actually emulate basic dopamine cell signaling is evidently what is responsible for the reinforcing effects. So it's not just increasing dopamine, but doing it very abruptly in a way that mimics and emulates basic dopamine cell signaling. So that's very nice in terms of our understanding of the reinforcing effects of drugs. But how does that in any way help us understand drug addiction? And why the question? Because anybody, whether you are addicted or not, if you get exposed to a drug of abuse, will produce these very dramatic increases in dopamine. And that will not explain, therefore, because whether you're addicted or non-addicted, you're going to get this pharmacological effect. Why is it that a person that's not addicted said, well, maybe I like it, it feels good, but that's it, versus someone else who's addicted that get the high and immediately have this intense drive for wanting more, which we call in the field craving, the craving for the drug, which is actually very difficult to control. So when you take a drug, though, you are emulating this dopamine cell system, basic dopamine cell system, but you are producing a massive disruption of dopaminergic pathways because you are inundating them. I said the basic dopamine cell system is very short-lasting, whereas you're taking these drugs that are just flushing the whole dopaminergic pathway. So as a result of that, it is not just affecting, while the basic dopamine cell activity, increasing it, is what's driving the reinforcing effect. At the same time, you're going to be increasing the concentration of dopamine overall the whole system. And that is believed to initiate the neuroadaptations that result in drug addiction. And what are those neuroadaptations? For many years, people were hypothesizing that people took drugs because the dopamine system, the limbic system, the reward system was involved. So it was a disruption, a disease of the limbic system. But it so happens that dopamine plays a role that goes way beyond just reward and reinforcement. Dopamine is crucial in modulating, very important for a wide variety of everyday activities and survival, how our prefrontal cortex works. This is the area here. And it is the prefrontal cortex that allows us to exert judgment. It's the one that enables us to exert, crucially involved in abstract thinking, crucially involved in our ability when we're placed with something that we want to do, like eating a chocolate that I shouldn't eat, to tell me, no, don't do it, and enable me to exert inhibitory control and not eat that chocolate. So that's what psychologists have called executive function. So the dopamine system is crucial for the proper function of the prefrontal cortex. Through the reward system, the dopamine system is crucial in order to enable motivation and drive. And finally, an area that is very, very relevant, that's much less recognized by people that are not on the field of substance abuse, is that the dopaminergic pathways are crucial for memory and learning. So when you are dealing with drugs that are going to be creating neuroadaptations on the systems that are regulated by dopamine, because they are not limited to one particular area of the brain, they are distributed widespread, then you start to question, well, what is the consequence of repeated drug administration on the function of these systems that are modulated by dopamine, in this case, through tonic dopamine cell activity? And so that's where we started in terms of inquiring the system vis-a-vis what are the disruptions on the brain of a person that's addicted. And our first strategy was to look, is the dopaminergic system, number one, affected, disrupted by chronic drug administration? When the person is not taking the drug, because I know that they are taking the drug, it's going to be activated, hyperactivated. But when they're not taking the drug, and if there is such a disruption in the dopaminergic pathway, how does that affect the way that the brain is functioning? So it's disrupted if I don't understand what are the consequences, so how does that help me in any way? So our first set of studies was actually to monitor the dopamine system by measuring the expression of the dopamine D2 receptors. And these receptors are crucial in terms of signaling reinforcing responses to drugs, but the dopamine D2 receptors are also involved in the proper function of those circuits that we're discussing here. There are other receptors too involved, particularly the dopamine D1 receptors, but the dopamine D1 receptors has been much less investigated. At the same time, since we were asking the question, well, what are the consequences of disruption of dopaminergic of receptors, which are crucial for transmitting the signals in brain function, we were measuring brain glucose metabolism. Because glucose utilization in the brain is an indicator of activity. And when there is disruption in activity, this is a very, very early biomarker of impairment of neuronal activity. And for example, using FDG, clinicians now can do early diagnosis of Alzheimer's disease before you actually see the symptoms. The same thing with Huntington chorea. And this is because you start to see, for example, in the case of Alzheimer's disease, decrease in metabolism, metabolic activity of glucose consumption in areas of the brain like the hippocampus or the precuneus. So we were studying cocaine abusers, measuring D2 receptors, and at the same time measuring brain glucose metabolism. So what did we see? Was the system disrupted vis-a-vis dopamine D2 receptors? And the answer is yes. We've done many, many studies, and similar findings have been replicated by other laboratories using different radioligands, using different technologies. Also in laboratory animal experiments, when you compare animals that self-administer versus not, dopamine D2 receptors is one of the variables identified as involved in the differences. So these are normal controls for different types, compared against different types of people that are addicted. And what we and others have found is in cocaine abusers, reductions in dopamine D2 receptors. In methamphetamine abusers, disruption in dopamine, decreases in D2 receptors. In alcoholics, decreases in dopamine D2 receptors. And these too have been found in postmortem studies. So this is very consistent. Heroin abusers, decreases in dopamine D2 receptors. So it's not something that is specific for any one type of addiction, but across a wide variety of drug addictions, we and others have found reductions in dopamine D2 receptors. Now, of course, the question is, well, so what? I mean, how do I know that this in any way is associated with a vulnerability for taking drugs? And that's one of the issues of why, and we were discussing earlier, the importance of translational centers. The ability of getting information, not just from the laboratory animal and bring it into the human, but as importantly, to get information on the patient population that you're studying and then test it in the animal. And why am I bringing this here? Well, one of the ways that I could actually determine that having low levels of dopamine D2 receptors make you more vulnerable to taking drugs is if I could do an intervention in these addicted individuals that led to an increase in the expression of these dopamine D2 receptors. If they were causally linked, then I should be able to demonstrate that that decreases their drug consumption. And then I would have a mechanistic association, causally linked. But I don't know how to do that in humans. Currently, we really do not have a way of increasing dopamine D2 receptors other than blocking them, which defeats the purpose. But we can do it in animals. So why not? We have plenty of animal models for drug self-administration, and that's what we did. Our first studies, we've now done 3 independent studies, 2 on alcohol and 1 on cocaine, with similar results. We make an animal self-administer alcohol in this case, and when the animal readily self-administer alcohol, we do an intervention to increase dopamine D2 receptors, and then we ask the question, does alcohol drinking go down, or cocaine drinking? Well, this is the data. The first study was done on Sprague-Dawley rats, and the way that we increased dopamine D2 receptors was using an adenoviral vector in which we inserted the dopamine D2 receptor gene, stereotactically injected it into the nucleus accumbens, and then measured the expression in the nucleus accumbens of the dopamine D2 receptors, and at the same time measure the behavior of self-administration of alcohol in this case. And what you see here is the increase in the expression of dopamine D2 receptors in the nucleus accumbens, as a function of time since injection of the vector. And we see approximately a 50% increase in the expression of the D2 receptors for day after administration. With the adenoviral vector, this is short-lasting, and by day 10, it's back to baseline. But we can inject the animals again, which we do at day 20, and again, the receptors go up. So under these conditions, the question was, well, what happens to the behavior? And this is a study on alcohol, but the same thing we got with cocaine. And what we saw was when you increase the receptors, in this case 50% overall, you see a dramatic reduction in alcohol intake, approximately 70%. And as the receptors go back to baseline, the drinking behavior subsumes back to baseline. And when you actually inject the adenoviral vector again and you increase the receptors again, the drinking behavior goes down. And here in blue are animals that were injected with the adenovirus, but there was no gene inside to control for non-specific effects. And in these animals, there's no difference in drinking behavior, documenting indeed that having low levels of dopamine D2 receptors makes you more vulnerable to taking drugs. But what is intriguing is, and again, I think I always say this, particularly for younger investigators, look at the data. The data is actually really the source of inspiration. Because in my brain, what it's basically telling you here, it didn't block it. The animals were still drinking. So it was fascinating to me that what it's doing is decreasing the amount of drug that the animals are consuming. But that's what addiction is all about. So if you take alcohol, for example, if you're a male, what becomes problematic, alcohol abuse of alcoholism, and when you start to drink 5 or more drinks a day. If you're a female, 4 or more drinks a day. So whereas drinking 1 glass of wine is not in any way problematic per se. So it appears that really what the low levels of receptors in the striatum are enabling, facilitating, is that compulsive administration of high quantities of drugs. And we get exactly the same finding with cocaine. The animals still administer some cocaine, very, very low levels. And when you look at the literature in terms of primate or rodents that are not manipulated genetically, you see the same process. Low levels of receptors in striatum are associated with consumption of higher quantities of drugs. Now, this is that association was eloquently demonstrated by Trevor Robbins and his group in a series of 2 experiments that were published in Science, in which he actually, his first study, he was looking at impulsivity, and he was interested on impulsivity because impulsivity is one of the factors that is linked as a risk factor for drug abuse in humans and in animals. And what he showed in this study was he divided animals that were non-impulsive and then animals that were impulsive. And he looked basically dorsal and ventral striatum non-impulsive, dorsal and ventral striatum impulsive. And what he reports is that the impulsive animals have a lower levels of dopamine D2 receptors in the striatum, both in the dorsal and the ventral striatum, which is where you have the nucleus accumbens. So just like the drug-addicted people, these impulsive rights have lower levels of D2 receptors. Then in a subsequent paper, what he did was he divided the animals into 2 groups, those that were impulsive versus those that were the impulsive versus the non-impulsive. The impulsive, we now know, have low levels of dopamine D2 receptors, and the non-impulsive have high levels of dopamine D2 receptors. And he puts them in a paradigm to determine whether, if they have free access to cocaine, will they self-administer it compulsively. And it's a paradigm, an animal model, that investigators are using it to understand vulnerability for drug addiction and compulsive drug intake. And what he reported was that those animals that were impulsive, which you actually see here on the scores, were the ones that were much more likely to administer cocaine compulsively. Thus corroborating to a completely different experiment that perhaps one of the mechanisms by which low levels of dopamine D2 receptors in the striatum in increasing the vulnerability to drug intakes is by favoring this compulsive pattern of administration. Now, of course, the big challenge now is how do you revert those changes in a human being? Now, the other question that comes around is why? Why is it that low levels of dopamine D2 receptors is resulting in this phenotype of impulsivity and compulsivity? Why? And you can sort of leave it to the striatum, the dopamine D2 receptors. Or you can actually try to inquire in the human subject, well, how does that affect the work, the way that the brain, the human brain works? And that's where the next side of the second chapter of those particular studies went in our lab. We were measuring brain glucose metabolism in those cocaine abusers, in which we were measuring dopamine D2 receptors. And these are just images to familiarize you of brain glucose metabolism on 3 different levels of a normal person and a cocaine abuser that has been detoxified, so they are not on any effects of cocaine. They have been clean at least 2 weeks. And you immediately see that there is decrease in glucose consumption because this is your scale. And the effects are actually most pronounced, interestingly, in areas in the brain that nobody had paid any attention until we started to report these findings with brain imaging. Because again, we were, like everybody else, interested on the limbic brain. So when we started to identify that in addicted people, the areas that were most affected vis-a-vis glucose consumption was not per se the limbic brain, but the prefrontal cortex, it was a big surprise because the prefrontal cortex had been traditionally thought of involved with higher levels of cognition, but not with limbic operations. But the data is the data, and that's always either. If you don't want to believe your data, then because it doesn't fit your hypothesis, then don't even bother to do the experiments. And look at the areas where this was very dramatic was single-age arrows, control cocaine abuser, and in this one, orbital frontal cortex control and cocaine abuser. And this finding was so strong that, and actually we've replicated it, and now everybody and their neighbor has replicated this, and nobody questions it. But when I was first showing this, no one believed it. And what was more intriguing about it was not just that there were decreases in metabolic activity in these frontal areas, but when we inquired the system, and again, no preconceived notions, it was a fishing expedition of fishing expeditions. Not that I wrote it like that on the grant. But my question was, I have these measures of dopamine D2 receptors in the striatum of normal controls and cocaine abusers, and I have brain glucose metabolism. I want to know, I want to inquire, I have the data there. Where are the reductions in dopamine D2 receptors in striatum associated with glucose consumption in the brain? So why should I not go fishing since I have absolutely no idea? I was, of course, hypothesizing that it would be in the limbic brain. But I look at the whole brain, and what we found was actually a big surprise. First of all, our hypothesis that it would be the striatum, because we are measuring D2 receptors in striatum, would be associated with decreases in metabolism in striatum. No, not at all. We look at the whole brain, and the areas, when you look at the whole brain, you have the problems of statistics because you have to control for multiple repeated effects, so you don't have false positive. So when we did a Bonferroni correction and then determine which areas were there a correlation between decreases in D2 receptors in striatum and decreases in metabolism, it was specifically these areas that I have here in white. Single-age arrows, dorsolateral prefrontal cortex, and orbital frontal cortex. And in red, I'm showing the areas where the correlation was the strongest. And both of them are consistent. We've replicated this so many times. And always the same effect. You have lower levels of dopamine D2 receptors in the striatum. You have decreased metabolism in the orbital frontal cortex, decreased metabolism in the anterior single-age arrows. And this is data for 2 of our studies, cocaine abusers, methamphetamine abusers, low levels of dopamine D2 receptors, low metabolism, orbital frontal cortex, low levels of dopamine D2 receptors, low levels of metabolic activity in the orbital frontal cortex. And you'll ask, well, what does this have to do with the compulsiveness and the data from Trevor Robbins? Well, if you damage the orbital frontal cortex, or if your orbital frontal cortex is disrupted because you have obsessive compulsive disorders, the characteristic, I already told you, obsessive compulsive disorders, is you engage in compulsive behaviors. And indeed, one of the first findings in the imaging literature of psychiatric disorders, that was patients with obsessive compulsive disorders have decreased activity in the orbital frontal cortex when they are not in the rumination behavior, or hyperactivity when they are going through their rituals. And to be honest with you, since I had never really paid much attention to the orbital frontal cortex until it started to smile at me in my studies, And there was no one studying orbital frontal cortex in drug addiction. And I am an imager. I was fascinated by the obsessive compulsive disorder. And I remember my colleagues, the psychiatrist and the chairman of the department, Fritz Ken, stands up and says, Nora, are you implying that obsessive compulsive disorders and drug addiction are the same disease? And I said, no, Fritz, I'm not implying that. Basically stating that we only have one brain, and there's so many ways that the brain can get disrupted, and that there is a common neurocircuitry, and that that disruption of the dopaminergic pathways is resulting in a dysfunctional orbital frontal cortex that then emulates that component of the OCD, the compulsive quality of the behavior, the obsessiveness with the rituals. The neurobiological underpinnings that lead there are distinct. But actually, curiously, and that's how I'm going to just an anecdote, because Kat is here and she doesn't know this story, but that's when I started to very much be very intrigued by the serotonergic system. Why? Because one of the treatments that is used for the treatment of obsessive compulsive disorder is serotonergic enhancing drugs. So it followed in my brain, well, why not target serotonergic agents in order to overcome that disruption of the orbital frontal cortex, which is loaded with serotonergic terminals? There are several problems there. So you'll say, well, haven't they tried them? And yes, they have tried them. The problem is that the combination of the main molecular compounds that we have for increasing serotonin, which are the SSRI, or serotonin transporter inhibitors, which are antidepressants, and you use to treat OCD, when combined with cocaine, are toxic. And so you cannot really increase the doses at levels that would be relevant. So that whole question has not really been properly explored. Though many psychiatrists, since I'm a psychiatrist and sort of you can practice, many psychiatrists do use SSRIs to help their patients stop taking drugs. But it's an area that obviously requires more looking into. The other aspects about it is that findings like this one then bring to light, well, how does this fit into the process of drug addiction? In animal models, and again, the beauty of being able to go back and forth, if you damage the orbital frontal cortex in an animal, what you generate is a fascinating phenotype. You actually train them to press a lever in order to get a reinforcer. And an animal with damage of the orbital frontal cortex learns that. They learn that if they press, they'll get a reinforcer. Now, an animal that doesn't have a damage, of course, learns. But as the investigator goes on and decides, well, now, little rat, I'm not going to get you your food, the animal learns to stop pressing for the lever because it's no longer reinforcing. Presses the lever, there's no food, and so says, why should I do it? But if you damage an animal in the orbital frontal cortex, or if you can generate the same process actually by damaging the caudate, so clearly there is a very powerful connection that mimics the phenotype, whether you damage the head of the caudate or you damage the orbital frontal cortex, that animal learns to press the lever, but when you remove the reinforcer, the animal continues to press the lever again and again and again and again, even though it's no longer reinforcing. And that actually emulates very much what the drug addict will tell you. I'm a very curious person, so I do ask the patients, well, why do you take that drug? They can take you different things, but a very common answer that I get, they look at me perplexed and says, I asked myself that question because I don't understand it. The drug is no longer even pleasurable, but I just cannot control it. So this drive, which in many ways, if you think about it, is not dissimilar to what happens with a patient with OCD. They know their hands are clean. They know that. And yet they have to wash them again and again and again, even though consciously they know their hands are clean and they want to stop. So you have that similarity in terms of the clinical presentation is likely to represent the common disruption of these neurobiological substrates. Now, this is a very important system, the prefrontal cortex, executive control. The single-age gyros is involved with our ability to actually monitor errors. So if I am right now telling you something that is nonsensical, my single-age gyros will get activated, and that would enable me to correct. Now, if you have it not functioning properly, you cannot correct, and it also makes you much more impulsive. So disruption of the orbital frontal cortex promotes compulsiveness. Disruption of the anterior single-age gyros promotes impulsivity, an inability to shift your behavior when you become on errors. And you can start to then understand why the behavior of a person that's addicted is so problematic. Now, as complex as this is, it would still be sort of simple in a way to then say this is the neurobiology of addiction, but it doesn't stop there. It does not stop at all there. These drugs disrupt multiple systems. And this is actually what you have here, the reward system. So that was the executive control that is disrupted in people that are addicted. Well, why that reward? And this was relevant because I wasn't going to ignore it just by the fact that we were reporting this disruption on executive function. And for many years, it was believed that people were addicted, and many still believe that, because the drug is more pleasurable, more rewarding. Ergo, it produces an enhanced increase in dopamine. And this line of thinking has been fed in part by an animal model, which is very valuable. Terry Robinson described it, which we call sensitization, whereupon with repeated administration of amphetamine or heroin, you can increase the amount of dopamine in the brain. Sensitization. So that has led some, well, the person that is addicted because the drug is more rewarding, more motivating, more pleasurable, more dopamine. So, of course, when you think about it and you put that, that's perfect for a grant and a study section and great hypothesis. So we did that and says, we're going to study the reward system, and we postulate that people that are addicted to cocaine are going to have a much greater increase in dopamine when we give them a stimulant drug, number one. And number two, they are going to perceive the drug as more reinforcing. Hypothesis, I think that probably was the grant that I have got the greatest score. I think I got one like the 1.1 or 1.0, something like that. That's why I think that actually, and my hypothesis was absolutely wrong. I got the money, it doesn't matter. I didn't have the behavioral responses. The behavioral responses, it's a pity I didn't. Here, yes, hi, here. Here I have the behavioral responses. So this is a normal control, and we're giving them intravenous methylphenidate, which, as I say, cocaine abusers report to be very similar to cocaine, and they like it. Now, when you're reporting self-reports of high, our big surprise was that controls report the intensity of the high to be much stronger than cocaine abusers. And this actually has then been replicated for amphetamine, and interestingly has been replicated similarly in similar type of studies that are being done in individuals that have morbid obesity, in whom you would expect that the food per se would be more reinforcing, pleasurable, and were exactly contrary to what would have expected. They see exactly the same thing. It's not that they like it more. It's that they want it more. So both in the drug addiction and in the morbid obesity field, it has become clear that these two are, while they may be linked, are not necessarily the same. Liking or wanting. I like chocolates. I don't want them right now. And I may want something that I don't like. If you food deprive me for 1 week, I will eat a raw egg, which the idea just makes so disgusting to my brain, but I'm sure I will. I will want it. So the concept of the wanting is very dependent on the context, as it is. The liking is a more constant one. I like chocolates where I'm in China or Mexico or wherever. I want them sometimes and not the others. So that's where the big distinction is coming around. And drug-addicted people do not necessarily, in that sense of the word, like the drug more. Not at all. If anything, it appears that those descriptors of positive effects of the drug are lower. Now, what about changes in dopamine? Just look at that. Again, completely wrong. Normal control with intravenous methylphenidate, you see that decreases in the binding of raclopride because dopamine is going up and it's binding to the receptor, so the radioactive substance cannot bind. Look at this cocaine abuser. Placebo, IV methylphenidate. This particular subject has absolutely no change. And we did, of course, control for plasma concentrations. Now, look at when you do the average, in this case, 20 controls and 20 cocaine abusers. This is our model parameter that reflects changes in dopamine. That response of the cocaine abusers is less than 50%, less than half that of a normal control in terms of the drug ability to increase dopamine in the striatum. So the person that's addicted is not addicted because they are having an enhanced sensitized response to the drug. I'm sorry, the data is the data. Identical results were reported by the group at Columbia in cocaine abusers using amphetamine. And they again reported 50% lower increase in dopamine in cocaine abusers. We also taken these studies, and now we've done it in alcoholics to try to see if this phenomenon is specific for cocaine addiction or other addictions. This is actually, I'm sorry, I apologize. I'm not presenting you the data from the alcoholics, but I'll tell you about it because it's absolutely dramatic. This is a study that we have not yet published in active cocaine abusers, very, very recent, as I say. Again, 17 controls and 17 active cocaine abusers, and versus the other one was detoxified subjects. And what you see is a very dramatic, the decrease in the binding of the raclopride in the control subject, and this is the cocaine abuser, and you can basically see almost no response. And this, you see the same thing with the high decrease in the cocaine abusers, and look in the active cocaine abusers, the response is even further attenuated in terms of the drug's ability to increase dopamine. So it's a very striking that a priori you would have not predicted. And this is the alcohol data I do have, and actually not just in cocaine abusers. So we replicated in detoxified and active, and the group at Columbia has replicated it in detoxified cocaine abusers. This is our data with alcohol. The group at Columbia has reported identical results with amphetamine instead of methylphenidate. This data, just to get you an idea of how powerful it is, in the past, we used to just show you one subject, and obviously you select a subject that is illustrative, which clearly means one of your best subjects. This data is not illustrative, this is average. So now we are averaging 20 controls, all of their already modified, sort of changed to represent all of the modeling, to just represent the binding to the receptors. 20 images from 20 control subjects placebo, 20 control subjects after intravenous methylphenidate. You see that decrease in the binding. These are 20 detoxified alcoholic subjects. And in this case, we studied them at least 5 weeks after last use of alcohol because we wanted to be sure they were not under withdrawal. And look, for this is average, not one subject. It's not my illustrative example. This is the placebo. This is the intravenous methylphenidate. Basically, visually, you would say there's no effect. When you quantify, what you see in the dorsal striatum, half the response of controls. In the ventral striatum, where you have the nucleus accumbens, 70% reduction. And as for the cocaine abusers, a decreased behavioral response. So what we're seeing in drug addiction is not the sensitized response of an enhanced ability of the drug to increase dopamine, but a very blunted response. It does increase it. It's not zero. It does, but it's much less so. Now, again, does that make sense? A priori, with the hypothesis, the models that we have, no. Because it would mean, why then would a person take cocaine if they are going to have such an attenuated response? And again, this is again which gives you an incline about how perverse drug addiction is with the brain, and where the memory systems become very, very relevant. Now, what memory? There are many memory systems, and I'm going to just concentrate, and the three of them, by the way, one of them classically identified in the hippocampus, very important for drug addiction, but I'm not touching it. Another one, the striatum, very important. This one in the hippocampus enables you to determine what you had for breakfast. The one in the dorsal striatum is a memory system that enables you to create rituals. Why are these important? Because it enables you to go everyday activities without thinking about them. It's a memory system. Dopamine is very important. But the one that I want to concentrate is this memory system that involves the amygdala, and which we call conditioning. The amygdala is part of the limbic brain, and from the inception has been of interest for the substance abuse field, and for many years has been investigated, and it's understood that it's important. Very important for emotional reactions. The amygdala is connected to the orbital frontal cortex. The amygdala is connected to the nucleus accumbens. And is important, it's crucial. If you damage the amygdala, you damage conditioning learning. Now, what is conditioning learning? Conditioning learning was first eloquently described by Pavlov when he demonstrated that in his dogs, which had been trained to be paired with a piece of meat with a sound, when the animals were exposed to the meat, they salivated. But when you pair that a sound with the meat, the animals started to salivate only with the sound. And that's what we call conditioning. Incredibly important for survival because it allows you to anticipate. So what's happening here is the animal, since they have paired those, the moment that they feel they are listening to their sound, they are anticipating the reinforcing effect of the food, and they are getting ready to get it. Well, the dopamine system is crucial in regulating that expectation and in monitoring whether if I expect, am I getting or not. Pavlov did not have PET cameras, so he couldn't go in and see inside the brains of his dogs what happens. But we do have PET cameras, and we can replicate those experiments. And in animal experiments, investigators have done that with cocaine, and looking at whether the dopamine system in the nucleus accumbens is involved in these responses of conditioning. And again, I repeat, if you damage the amygdala, you will not get these conditioned responses. But the question is the dopamine system in the nucleus accumbens involved? What they did, this is a study by Phillips et al. Using voltammetry, and he took two groups of animals, purple animals, green animals. Green animals, we basically just expose them to the sound repeatedly. Purple animals, the sound was repeatedly paired with cocaine. Cocaine and sound came together. Cocaine and sound came together repeatedly. Then they bring them back to the laboratory. There's not going to be any cocaine, but we're going to measure dopamine in the nucleus accumbens. And we're going to expose them to the sound, just like Pavlov did, and salivation occurred. Expose them to the sound, and let's look at what happens to dopamine in the nucleus accumbens. And what Phillips et al. Show, and others have shown the same thing, is that when you expose the animal to the auditory cue and measure dopamine in nucleus accumbens, there's no change. It doesn't do anything. But when they take the purple animals where they have been exposed that sound with cocaine, dopamine goes up in the nucleus accumbens. So this is emulating the pharmacological effect of cocaine, even though there's no cocaine. So the animal is activating the dopaminergic pathway in the nucleus accumbens. And that, in animal studies, the magnitude of this increase is directly linked with the motivation of that animal to press the lever in order to get cocaine. So here you have it, the conditioning responses mediated through the amygdala, which enables you to make those associations, through their connection of amygdala and nucleus accumbens, and by the way, the orbital frontal cortex is also involved with this, but I'm not getting into that, enabling glutamatergic signaling to increase dopamine self-firing, basic dopamine self-firing, that increases dopamine in the nucleus accumbens. Does this happen in humans? Well, we did the experiment. Why not? We can measure dopamine changes. We're not going to give any drugs. We're going to, I'm showing you the average images. These are 18 active cocaine abusers studied with raclopride, and these are average images across all of them. No drugs whatsoever. So that drug, intravenous methylphenidate, inactive cocaine abusers barely produce a change. So, but I'm bringing them under two conditions. One of them, I show them a video of nature scenes, all beautiful and calming. And in the other one, I show them a video of people preparing and administering cocaine. And I measure the changes in raclopride binding. And here's what you have. Control video, the high binding in the striatal area, cocaine Q video. And you can clearly visually see that the binding is going down because these cocaine cues are increasing dopamine. Moreover, the increases in dopamine produced by this cocaine cue are directly associated with a desire for the drug. And then you start to see the process. What has happened is you shifted. On the one side, the drug itself has lost its ability to increase dopamine, but that ability is shifted to the conditioned stimuli. Now, what are those conditioned stimuli? Any stimuli that has been associated with taking the drug, which could be the pipe that a person uses. It could be a person, which with the person takes drugs. It could be a neighborhood where they acquire the cocaine. In the case of nicotine, this is even much worse because people smoke. They used to smoke more throughout their everyday activities, so everything gets conditioned, and everything will trigger these increases in dopamine that will translate into the brain, expect the reinforcers, and activate the motivational drive circuit that engages the person into doing the behavior to take it. These responses are actually very fast and sometimes not even conscious. In our case, we do see a relationship between the magnitude of the changes and the craving, and identical results actually were reported 2 months later by the group at Johns Hopkins led by Dean Wong. So taking this information, we can start to get a landscape of how, through the eyes of dopamine system, many of these circuits, as I mentioned, clearly as it relates to the orbital frontal cortex and single-age gyros, serotonergic systems are very important on regulating it. But this model came through the eyes of dopamine. Doesn't mean that other systems are not involved. You know, and glutamatergic pathways are crucial for these conditioned responses. Now, we take it, simple model. We know it's more than this, but I like sometimes simplicity. This is a normal person where the brain circuits are not in conflict with one another. Let's say my brain. My brain, I always like to think I have a big prefrontal cortex, so I said it works fine. That's not necessarily the case, but I like to certainly say, yes, my prefrontal cortex works well. I am subject, because I'm hardwired to like things that make me feel good. Anyone does, and I like chocolate, so that's one of the things that makes me feel good. And since I do know chocolates very well, I can see a chocolate, and Kathy was sure to provide me with all sorts of conditioned stimuli when I get there, of all types of chocolate. So I am saying, aha. Immediately I see the chocolate, I want the chocolate, and I'm hungry, so that increases the saliency value. But I'm saying in my prefrontal cortex, no, it would be really inappropriate to eat the chocolate because Kathy has not even offered it to you. So it's not proper behavior. So my prefrontal cortex says, no, 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 Nora, don't take it. And I inhibit it, and I don't take my chocolate, and I stop. No. That's the way that I control. Do I always control so well? No. And this is an important aspect, because we all, and again, the notion of wanting is context dependent. So there are going to be circumstances where that context is going to drive us to a more impaired prefrontal cortex, which is when? When do we have impaired prefrontal cortex? When we are frustrated or angry. And that's why you're much more likely, certainly I am much more likely to eat the chocolate when my plane has been canceled or delayed by 2 or 3 hours. So I may have been saying, no, I'm not going to eat it. And they say, we're sorry to inform you, but your plane has been your plane has been canceled, and immediately I go and get a chocolate chip cookie. I cannot control it. And there is really elegant work right now trying to identify under how all of us, under certain conditions, can have disruption on self-control, and why. But overall, for the most part, we're able, of course, to regulate our drives and desires and to make judgments and decide, is it worth it or is it not worth it? In a person that's addicted, the story is very different, because you saw that prefrontal cortex is not working properly. Single-age arrows, orbital frontal, dorsolateral prefrontal cortex. The value of the drug as a reinforcer is highly conditioned, and it's actually, you saw that decreased function of the reward system that was evident even with a drug. What other people, including our laboratory, has also been showing is that that's not just blunted responses to drug rewards, very blunted to natural reinforcers, whether it's food, sex, or money. So the limbic brain of a person that's addicted to drug is much less reactive to natural reinforcers, And so even though there is an attenuation of the reaction of the drug, in our brain, in our life, everything is relative. So drugs have no competition. It's basically the main drive for a positive reinforcer. So suppose that this is a person that is an alcoholic and sees the alcohol, and they want to drink the alcohol. They get the conditioned responses actually invoke central and peripheral mechanisms, and when a person is exposed to a conditioned cue, their heart rate goes up, they start to salivate, they start to sweat. And so they see the alcohol, they want to drink it, and they have this very powerful wanting response driven by this conditioning activation of the pathway. And they say, well, no, I have to give a lecture. That would really be bad. Imagine in front of the pharmacologist being intoxicated with alcohol. So that person does not want to drink, but the prefrontal cortex is not working properly. Now, those of you that have followed the work of Tony Grace or Leduc, which very different investigators looking at different things, they come to a common point that actually illustrates what happens in a situation like this. I said the amygdala is extraordinarily important in these conditioned responses. You get conditioned stimuli, the amygdala gets activated, that sends the signals to the nucleus accumbens. But the amygdala is also very important in emotions. So that emotional reaction, when it's very intense, does something that is very important for survival, but very detrimental for drug addiction. When the amygdala gets activated because there's something emotionally very, very important, here comes a tiger. I love tigers, I love animals, but I think that if a tiger went coming up there towards me, I will not stop to think, is it wise to try to basically pet that little tiger, or should I run? So it's in my best interest not to lose that 200 milliseconds that would take me to see it right or wrong, but to actually immediately run. So under those circumstances, with high level of activation of the amygdala, the prefrontal cortex gets disconnected. So even though you start with, you didn't have much of a chance because it was not working properly, when the amygdala is hyperactivated, the prefrontal cortex gets disconnected. And that's the way that you control your ability to say, no, I'm not going to do it. So you remove the brakes. So even though you may want to say, I want to brake, there's a cat crossing the street. I love cats. But I'm doing this to be dramatic. The least thing I want to do is to hurt a cat. But if I am driving and I have no brakes, even though I consciously want to brake, I won't be able to do it. And that's, again, the concept of the neurobiology, why consciously, through cognition, cognitive operations, enable us to control our desires. But those cognitive operations depend on the prefrontal cortical circuitry. And if that prefrontal cortical circuitry is disrupted, which will be exacerbated by these strong conditioned emotional responses, you will actually lose the ability under those conditions, in that context, to control the behavior. And the person is not able to stop. Now, what happens? They cannot stop, so they take the drug. And they are taking the drug. They are conditioned to the drug. So dopamine is already going up before the drug gets into the brain. And that activates the motivational circuit, and they want more drug. And that activates the motivational circuit. This is completely disconnected. And so they go, and 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 they go. This is a compulsive behavior driven by the improper function of prefrontal cortical circuitry that is actually at the essence of the disruption in the process of drug addiction. Now, when you get data like this, when you say, okay, that gives us clearly a glimpse about why the behavior is so disrupted in people that are addicted. But even more importantly, it provides us with strategies in terms of intervention for the treatment of drug addiction. Why? Well, again, I always tend to be described, and Kathy was saying, that's just the glass half full. I take it as an optimist. The fact that multiple systems are involved in drug addiction, of course, makes our task harder, but it can also be utilized to manipulate it. Why? Because you might have a person whose prefrontal cortex is very disrupted, and you may be able to intervene by strengthening this element and then help buffer the system. So, and this also gives us an explanation by why a person that's addicted to drugs during certain periods of their life 2, 3, 4, 5 years, they do very, very well. And then something happens. They get usually a stressor. Their spouse divorces them, or they go bankrupt, or a stressor. And that leads them to the compulsive intake, because the ability of buffering of that system gets disrupted. In this case, the emotional reaction from the divorce or whatever basically makes this connection disrupted. So the ability of having different nodes at which you can enter to buffer the system and to maintain a better function, to me, is a strategy that we should be doing with drug addiction. People that treat cancer don't stay and say, okay, I'm going to use this chemotherapeutic agent. People that treat cancer treat it aggressively and use every single armamentum that they have in order to improve the outcomes. There's no reason why we shouldn't be doing the same thing with drug addiction. So multi-pronged approaches to strengthen prefrontal control, multi-pronged approaches to interfere and inhibit or erase those memories of conditioning, multi-pronged approaches to enhance the saliency of reinforcers that are other than the drug so that they are competing. And strategies, of course, at the same time, that decrease the reinforcing effects of drugs of abuse in order to be able to help that person from going go to be able to stop. And with that, I stop my presentation. But before that, I do want to thank my colleagues at Brookhaven National Laboratory, with whom this work would have not been possible, in particular, Joanna Fowler, who's the head of the PET program at Brookhaven National Laboratory, Jean-Jacques Wan, who's the nuclear medicine physician responsible, along with Frank Tell and the neurologist, for doing these studies. And of course, the radiochemists, all of the rest of the radiochemists that are able to label these compounds. And I always thank the generous support of the National Institute on Drug Abuse, and I thank you for your attention.